I think that
Thank you guys for your patience. So yeah, as you mentioned, we have the slides up now. It's recording, so you can look back at this lecture after I finish. So again, just kind of recap from previous lecture, you guys looked at. We talked about glycolysis. Again, this is occurring in the cytosol, and again, this was an anaerobic state, so no oxygen yet. Now we're going to talk about the next step that this metabolism pathway is going to take into the mitochondria. So just to illustrate kind of where we are, again, the mitochondria, it has several different layers. So in dark orange here, you can see the outer membrane. And then here, kind of within this invagination, which we call cristae, this is the inner membrane. And in between these two membranes, this is called the intermembrane space. So in this gray area here, this is the mitochondrial matrix. This is where a lot of what we call the TCA cycle is going to occur. So this is where, as you can see with this closed loop, we're going to have a lot of these reducing equivalents in the form of NADH and FADH2 being generated. And these are going to feed into this intermembrane space, where we're going to have a lot of these protons labeled as HPLUS being used to form this proton gradient that is ultimately going to give us sufficient ATP. And we'll talk about that in more detail as we move on in the lecture. So I also want to focus your attention on the upper right-hand corner here. So this is also, so we talk about triglycerides. If you guys remember, these are fat storage molecules. And what's important about this is that, for one, they're also occurring in the matrix. But second is that they're also going to be broken down and feed into the TCA cycle. So just keep that in mind. We'll talk about fat oxidation and breakdown of fats again in upcoming slides. Also, just a reminder, if you guys have any questions, I'm really trying to make this interactive. So, your professor's teaching style, if you have questions, feel free to raise your hand or just speak up. I'll be happy to answer this. So as I mentioned, we're taking the next step in this glucose metabolism now with aerobic respiration. So here, this really boils down to two things. So first, we have the TCA cycle. We call it the tricarboxylic acid cycle, but we also call it the Krebs cycle, which was named after its founder. We also refer to it as the citric acid cycle, and we'll talk about why we call it that in a second. So really with the TCA cycle, you want to be focusing on two things. First, as I mentioned, we're generating these reducing equivalents. So one of these is NADH. And second, we're also going to be talking about how we're using amino acids, so the monomers of proteins, as well as other things like fats to incorporate into these metabolic pathways. So next, we're also going to be talking about oxidative phosphorylation. So if you remember, I mentioned that this is really occurring within the intermembrane space. Also, just keep in mind that we also call this the electron transport chain. And the reason for this is because, if you remember, these reducing equivalents are going to feed into this process. And what we're going to use is these electrons to create this proton gradient and give us this ultimate ATP synthesis. Again, just keep in mind that two roles of this oxidative phosphorylation is, one, as I mentioned, the proton gradient, and second, how we use that gradient to then generate ATP. All right. So now the TCA cycle. So here you can see on the right, this is a brief overview of what it looks like. You can see it's a closed loop, as opposed to what you guys talked about in the last lecture with glycolysis, more of a linear top to bottom mechanism. Here in this upper left corner right here, you can see we're starting with pyruvate. If you guys remember, this was the product of glycolysis. And sort of in order to prime this pyruvate to enter the TCA cycle, first what we need to do is we need to work with this pyruvate dehydrogenase complex, so PDH. And what this is doing is it's taking this pyruvate molecule and it's oxidizing it. As it oxidizes it, we also see oxidative decarboxylation. So what I mean by decarboxylation is we're seeing the removal of CO2. And at the same time, we're also generating one NADH molecule. And this is giving us one acetyl Coenzyme A, this 2-carbon molecule. I'll also add that as we move into the TCA cycle, we're also going to focus on some key steps. And we don't expect you guys to memorize all the intermediates, all the enzymes, but if you're interested here, it conveniently lists them. But I will, again, just focus your attention to some of the key steps, and we'll go over those in the coming slides. But first, I think it's important to talk about Coenzyme A. So if you remember from what we just mentioned with pyruvate being converted to acetyl Coenzyme A, this is a part of this acetyl group, and its main function is delivering these acetyl groups to oxaloacetate, one of the first intermediates of the TCA cycle. So just to give some background on the structure of Coenzyme A, you can see it resembles a lot of what you guys are already familiar with. So if you remember with DNA, we have the ribose sugar, highlighted in light blue right here. And we also have a nitrogenous base. This one is the adenine, but you guys also remember cytosine, thymine, and guanine. Here it just happens to be adenine. And then we also have this pyrophosphate bridge. So again, very similar to what you guys have learned before, but you'll also notice that it has this pantothenic acid. What's interesting about this is it's actually a derivative of vitamins. So this is the derivative of vitamin B5. And then linked to that, we also see the sulfhydryl group represented in SH. And if you're interested in knowing, like again, what this acetyl group is structurally, you can see it highlighted here in light blue. So it's essentially a carbonyl with a methyl attached, and that's represented by CH3. So again, just to summarize what Coenzyme A is being used for. So one, it's helping the entry point for aerobic respiration. So it's delivering these acetyl groups to oxaloacetate. But it's also the product of fat oxidation, and it's the substrate for fat synthesis. So it's sort of a precursor performing a lot of the fats that we'll talk about. And this will play a role in a lot of the dieting strategies we'll talk about towards the end of the lecture. All right, so now we just talked again about kind of how we prime pyruvate into acetyl Coenzyme A. We generated one NADH, we released one CO2 molecule, and now acetyl Coenzyme A is ready to perform its job of delivering those acetyl groups, those 2 carbons, to this first intermediate oxaloacetate. So I'll add that as we're adding these acetyl groups, this is a very exergonic process. So we have a large quantity of energy being released in this process, so it's irreversible. And it's a hydrolysis reaction, so we're using water to catalyze this reaction in the formation of citrate. What's unique about citrate is that this is a carboxylic acid, and so it has 3 carboxylic groups. I'll also point your attention to, as we're moving from each step of the TCA cycle, it's important to kind of focus on which carbons we're losing, because you remember from earlier, sort of a hallmark of this cycle is the release of CO2. So to kind of keep track of which ones we're losing, you can see that they conveniently highlighted carbons in different colors. So right here in pink, these are the carbons that came from acetyl Coenzyme A. And conveniently in gray, these are the ones that were originally in oxaloacetate. So as we move from step to step, you can see which ones are being lost and which ones are remaining. All right, so now we left off with isocitrate. So the next 2 steps are pretty important. What's going to happen is that, as you can see, we have the generation of these NADH reducing equivalents. So what enzyme is catalyzing these reactions? These are dehydrogenase reactions. So again, you don't have to focus too much on the names of these enzymes, but I'll mention them just for completeness. So as we move from isocitrate to alpha ketoglutarate, the enzyme here that's catalyzing this reaction is isocitrate dehydrogenase. as we move from alpha ketoglutarate to succinyl coenzyme A, this is catalyzed by, as you can imagine, an alpha ketoglutarate dehydrogenase. So both dehydrogenase reactions that are giving us an NADH molecule, but also releasing the CO2. So also a decarboxylation reaction. And what's important here is that as we arrive at succinyl coenzyme A, what's happening here is that we're generating a GTP molecule, so a guanosine triphosphate. And what's important about this reaction is that first we see the cleavage of the thiol coenzyme A group right here. And again, this is releasing a large, significant amount of energy that is being coupled to the substrate level phosphorylation of ADP. So as I mentioned, the product of this reaction here is GTP. But what's happening is that we see what happens is that there's these dinucleotide kinases that eventually take a phosphate from GTP and transfer that to an ADP to form one ATP. And again, because this cleavage of this thiol ester coenzyme group is so highly energized, we can use some of that energy then to facilitate the substrate level phosphorylation and generation of one ATP molecule. And again, just to kind of echo, as we're going through these steps, these last two were not only giving us this NADH, but also releasing CO2. And as I mentioned, it is important to keep track of which carbons we're losing. So in this case, you can see that we still have these two pink carbons. And so these are still being kept from acetyl coenzyme A. However, you can see some of these gray carbons are being lost. And so what's happening is that we're losing some of the original acyl acetate carbons. Just keep that in mind. Before I move on to the next step, does anyone have any questions? Go ahead and talk for a bit. Yeah.
I was going to ask, if the GTP may or may not be secondary ATP, what else would it use?
If it's not, if GTP is not used to generate ATP, what else would it be used for?
How could the cycle continue?
The cycle is not dependent on GTP. It's not dependent on GTP being a substrate to dinucleotide kinase. The important part is the production of succinate. It can, via that kinase, phosphorylate ADP and ATP, but it's not dependent, but is again kind of like it's advantageous to be able to generate that ATP during the process.
Anybody else have any questions of steps we've gone over so far? Okay, so now we've just arrived at succinate. Now the next step, we see another dehydrogenase reaction, so succinate dehydrogenase, which is giving us this fumarate intermediate. Now what's unique about this step is that, again, we're generating a reducing equivalent, except this time it's in the form of NADH2, so a flavin adenine dinucleotide. Next, I was going to redirect your attention to the final step of this TCA cycle. So after we arrive at malate, what's happening is that we have another dehydrogenase step with malate dehydrogenase. And what this is doing is not only generating another NADH molecule, but also giving us acyl acetate. So the first intermediate of the TCA cycle. And so you can imagine, as we arrive at acyl acetate, what's happening is that it's again awaiting its acetyl group so that it can continue the cycle. So now this is kind of the completion of the TCA cycle, kind of the run around the bend. So as I mentioned, with succinate dehydrogenase catalyzing that reaction and giving us an FADH2, I thought it was important to talk about what the structure looks like of this coenzyme. So similar to coenzyme A, it's similar in the sense that you have this ribose sugar, as well as this adenine nitrogenous base and the pyrophosphate bridge. But what's different is what's attached to that pyrophosphate bridge. So here you can see it's called a riboflavin. So what's interesting about riboflavin is that this is also a derivative of vitamin B2. And you can see also how it's being reduced and oxidized and how it's kind of going back and forth as we see the dehydrogenase reactions going on. All right, so as I mentioned, we just completed the TCA cycle. This is kind of a great overview of kind of what we used and what we got from this cycle. So just a reminder, we started with Coenzyme A. We have these oxidized equivalents here with NAD plus and FAD. And we have these ADP and PI orthophosphate ready to be phosphorylated to generate ATP. And then the products, if you remember, with the oxidative carboxylation reactions, giving us the release of CO2. And then again, sort of the hallmark of this cycle, the production or generation of these reducing equivalents. So NADH and FADH2. And again, that one step where we have the formation of GTP and then the substrate phosphorylation to give us an ATP.
Can you explain the sort of definition of the term reducing equivalents?
Yeah, of course. So what's happening is that you can see here we start with these coenzymes that are oxidized in this form. And as we have these dehydrogenase reactions, what's happening is that the substrates, so for example, alpha-ketoglutarate, they're being oxidized, and so they're losing electrons, and those electrons are being transferred to these coenzymes, and that's where they're essentially being reduced. That's what we call reducing equivalents.
Okay, so a reducing equivalent is basically just a substance that's been reduced.
Essentially, and the important piece there is that these are going to eventually be used in oxidative phosphorylation to transfer those electrons to these different electron carriers, form the proton gradient and stuff. Great question. And so then, also just keep in mind that as we just did this process of glycolysis, we left off with two pyruvates, right? And so what I just described is going to happen in shuttles. And so with these products, what's going to happen is that you have to multiply them by 2. Again, because we have 2 pyruvates, and so the cycle is going to occur in shuttles. So now just kind of taking a step back, thinking about how much ATP, NADH, and FADH do we have so far from glycolysis in the TCA cycle. We arrive at 4 ATP. So 2 ATP from glycolysis, 2 ATP from the TCA cycle. And then with NADH, we had 2 of these from glycolysis. And then we had 6 from the TCA cycle. Right, so that's 8. I'll pose a question to you guys. Do you guys remember where these other 2 NADHs are coming from?
That's when specifically when you're prepping pyruvate into
Exactly.
acetyl.
Good job. Yeah, so as you're priming with pyruvate and it enters this pyruvate dehydrogenase complex, remember we form acetyl Coenzyme A, and in the process we're generating our first reducing equivalent NADH. We're also releasing 1 CO2, and again we're forming acetyl Coenzyme A, which then feeds into the TCA cycle. Good job. And then lastly but not least, we have the formation of 2 FADH. So remember this is coming from succinyl Coenzyme A being catalyzed by this enzyme called succinyl synthetase and forming succinyl. And so now kind of taking a step back again, we see that we only have 4 ATP. So again, this isn't a lot, it's definitely not substantial, And so you might be asking yourself, okay, so how are we going to get enough ATP? And the answer lies in those reducing equivalents, right? So you can see we have 10 NADH and 2 FADH2 molecules. These electrons that they obtained in the TCA cycle are now going to be used in oxidative phosphorylation. And we'll talk about how that works. But also, again, I'll open the floor if anybody has any questions so far. Anything comes up. Okay, so now this is our introduction to the electron transport system. So again, remember this has two names, so oxidative phosphorylation and ETC. As you can see, sort of, first I'll orient you to where we are. So sort of at the bottom of this inner membrane, we have the mitochondrial matrix. So this is where we left off in the TCA cycle. You see right here, these reducing equivalents like NADH. And over here, you see the intermembrane space. And this is where we're going to find a lot of these protons being pumped across the intermembrane. I'll also point your attention to these different respiratory complexes. So within these respiratory complexes, again, we have, just to remind you, we have 4 of these. So complex 1, complex 2, complex 3, and complex 4. And within these complexes, what we find is these different electron carriers, like flavin mononucleotide, as well as these iron sulfur clusters. And what these are doing is they're transferring the electrons from the reducing equivalents. And as they do this, what this is doing is facilitating the movement of protons from the mitochondrial matrix to the intermembrane space. And what this is doing is creating a proton gradient. So you can see the aggregation of these protons in the intermembrane space, and that's creating an electrochemical gradient.
When the protons are moving up their concentration gradient, the energy to move them against the direction, I guess they naturally want it to move. So that comes from the dissociation of the electron from the intermediate substrate.
Yes, that's a great question. So actually in the next slide, we'll talk about how this actually works. So just kind of a preview, I guess, as these electrons move from iron sulfur cluster to iron sulfur cluster, what actually happens is you see a lot of conformational changes, and that allows, there's actually pores in these cavities that make up the complex that will sort of open and close that allow the protons to move into that space again, but against its gradient. So, as I mentioned, that's kind of how the electron transport chain is oriented. That's kind of how it's working. But to provide more detail on this pathway, we're going to center on complex 1. So what's interesting about complex 1 is that this is actually one of the more well-studied complexes. So this figure is from 2013. And what's interesting about complex 1 is it's actually in this L shape. So again, to kind of orient your attention to where we are, this L shape is pointing towards, so this vertical part of this complex is pointing towards the mitochondrial matrix. So this is the matrix right here. And then this horizontal part is the inner membrane, and this is kind of this would be the intermembrane space. And so to point your attention to the upper right, you can see here that this is the NADH reducing equivalent. So again, this is where we left off with the TCA cycle. What's happening first is that we see the transfer of these electrons from NADH to the first electron acceptor in flavin mononucleotide. And then sort of the next steps that we see is the transfer of these electrons to these iron sulfur clusters. And what happens as this happens is that we see this percolation that's depicted in these red arrows. And this moves down these cavities. Also to remind you that this complex is made up of these different subunits colored in with these different colors right here. And as she was mentioning, so how does this work? Like how do the protons move against the gradient into the intermembrane space? So what's happening is that as we have these electrons move into these cavities, there's actually pores here. And what they're doing is, so as we have these electrons moving through this, these cavities, depending on their oxidation state, they will either be able to bind the protons, or they will have less affinity and release these protons. And so here's an example. So before we have the electrons present in these cavities, you can see that they aren't able to bind to the protons. So they are closed off to the matrix. But as we see the movement of electrons depicted in these red arrows, you can see that this space opens and these protons are able to bind to these pores. So this happens as the electrons move to the cavities. Again, you see this space opening up, and that allows the protons to bind. And then you can imagine, so
One of here, very curious, what would happen if it transport system and the proton gradient and the complex, you have a lack of electron?
yeah, so if you have a lack of electrons, this would probably halt a lot of the mechanisms that we're talking about. You would see a lot of that proton gradient being formed. And I assume this is probably the case with some pathologies, some disease, right? Maybe a lack of.
Possibly, but I think the main takeaways, I think, as you said, I think pretty well, that in the absence of these electrons being provided by the reducing agents, NADH or FADH2, you wouldn't be having degeneration.
So again, just to talk about, again, these pores being open and closed based on their oxidation state, this is really how we see the movement of protons from the matrix into the intermediate. I know when I learned this slide, it was a little bit complicated, and I didn't understand it at first, so I'll leave the floor for any questions. You guys, if you didn't understand this mechanism. Yeah.
Just to sort of summarize, as the electrons are passed from the iron sulfide clusters to other cavities in this complex, that's changing the conformation of these different subunits and little clusters in the larger complex, which allows the hydrogen ions to sort of trickle through.
Exactly. That's a good explanation. So again, just to kind of summarize, so we have this percolation of these electrons from iron sulfur clusters and different coenzymes like flavin mononucleotide. And as this happens, we move from complex to complex. And in the process, we also see other coenzymes involved, like coenzyme Q here. This is involved with complex 3. Now bring your attention now to sort of this. Here we see that each complex is going to be delivering different protons, a different amount of protons. So with complex 1, you see that we're delivering 4 protons. This is because we have 4 cavities. This is a similar story for complex 3, where you also see 4 protons being pumped. But what's different is with complex 4, we only see 2 protons. And this again is because of the quantity of cavities. And in total, we have 10 protons. And so kind of ending the story, right, with the electron transport chain, again, we have this movement of electrons through these different complexes. And what happens is that they eventually arrive at the final electron receptor. So you guys remember from a previous lecture, you learned that this is oxygen. So oxygen is essentially this molecule with a high reduction potential. So it has the largest affinity in this context for electrons. And as the electrons move to complex 4, they are meeting oxygen and forming water. So this is sort of the end of that journey. And so, if you guys kind of noticed, we talked a lot about complex 1, complex 3, and complex 4. What we didn't talk about was complex 2. And we'll talk about why that is in the next slide. And so, here you can see complex 2. This is again embedded within the inner membrane. And the reason that we didn't talk about it as much in the previous slide is because it doesn't facilitate the movement of protons. So, big emphasis again, it doesn't facilitate the movement of protons. But rather, it still facilitates the movement of electrons, as you can see depicted in red arrows, complex 3 and complex 4. And as you can imagine, because also just to remind you that the electrons that it is working with, these are coming from the flavin adenine dinucleotide, so the FADH2. What's interesting is that if you remember, this reducing equivalent is coming from the succinate dehydrogenase reaction that we talked about earlier. Yes, the question.
For the second bullet point where it says 4 fewer protons moved, what is that in reference to?
Yeah, I was going to talk about
I'm sorry.
No, that's a great question. That's a great question. So yeah, this is a really important point. So because the electrons from FADH2 are not interacting with complex 1, they're sort of escaping complex 1. If you remember from the previous slide, we talked about how complex 1 is facilitating the movement of 4 protons. And so the electrons from FADH2 are going to contribute to fewer protons. And just to remind you, NADH2, these are interacting with complexes 1, 3, and 4. And so those are moving 10 protons. But FADH2 will only move 6. Great question. All right, so we just kind of talked about the journey of these electrons through this transport chain. We talked about how they are meeting oxygen, the final electron acceptor, in the form of water. And so we still left out part of the story with how we're generating this ATP. So this is the answer to that question. So if you remember from the previous slides, we also have this interesting looking purple structure here. This is F1, F0 ATP synthase. And it's sort of divided into 2 different compartments. So first, this F1 multi-subunit subunit protein. And then this core here represented this is F0. And this is within the embedded within the membrane. And then you also see this stalk, which connects these 2 compartments, so F1 and F0. Sort of the analogy that a lot of professors give, I remember when I took this class, they refer to it as sort of like a motor. And the reason for that is because what happens is, as these protons, again, remember, we pump these into the intramembrane space. And so what is happening now is we see those entering back into the matrix through this synthase. And as this happens, what we see is the rotation of this stuff 120 degrees. And if you're interested in how that looks, you can see in this sort of figure right here, it's represented in green. This is the stalk moving. And as it moves, what we see is the protons flowing through the synthase. And we see the drive of this ATP synthesis. So we see the phosphorylation of these ADP molecules into ATP. And again, just to emphasize, it takes 3 protons through this ATP synthase in order to rotate the stalk 120 degrees and drive the synthesis of 1 ATP molecule. Now, so we just talked about.
Good question. So do the electrons that the coenzymes carry, so do they both bind to oxygen to form water and then also facilitate the formation of ATP by traveling down the ATP synthase?
Yeah, that's a great question. Yeah, so they're both contributing to the movement of protons to the intramembrane space. Just remember that FADH2 is only pumping 6 protons because it doesn't interact with complex 1. But they're both contributing to that gradient. And so these are both going to come back to the synthase and contribute to the ATP synthesis.
Got it. Thank you.
Yeah, great question. Alright, so now it's kind of an overview of this oxidative phosphorylation. Remember, these first 2 points are just reminding you that because these electrons from FADH2 aren't interacting with complex 1, they're not going to drive the 4 protons, and so they're only going to be 6 total. And then kind of thinking about how much ATP we can generate from 1 glucose in this process of oxidative phosphorylation. So first we can focus on NADH. So as we have 10 NADH molecules from glycolysis and the TCA cycle, if you remember from these electrons in NADH, they can pump 10 protons into the intermembrane space. And so we can multiply 10 NADHs by 10, giving us 100 protons, right? If you also remember that it takes 3 protons to be pumped back through the ATP synthase to generate 1 ATP. So we have to divide this number 100 by 3, giving us 33.323 ATPs. But you see that we round down to 30 ATP molecules generated. Now the reason for this is because when we talk about the transfer of electrons from the leucine filaments to things like clade mononucleotide, this is not exactly a perfect transfer. And in fact, some of these electrons actually leak out and interact with oxygen, forming these reactive oxygen species, these ROS. And we'll actually talk about that in some slides later on in the lecture. Those will be actually pretty important with mitochondrial dysfunction. Now with FADH2, just a reminder, we left off with 2 of these. And because those electrons are only delivering 6 protons across the inner membrane, we multiply 2 by 6, giving us 12. 12 divided by 3, leaving us with only 4 ATP. So as a whole, ending with 34 ATP total from 1 glucose. So now this is kind of like the completion of these 3 metabolic pathways with glucose. Just a reminder, we have 2 ATP from the glycolytic pathways, so glycolysis. We have 2 ATP from the TCA cycle. And you have 30 ATP from oxidative phosphorylation via NADH and 4 from FADH2. So as a whole, we see 38 ATP from 1 glucose. And again, just kind of visualize the products and the reactants of this whole reaction. You can see we started with 1 glucose, C6H12O6. And again, remember this is an aerobic process, so we needed oxygen. That's why you see these 6 O2s here. And then of course we have the precursors for phosphorylation of ADPs, so ADPs are here. These orthophosphates. And then the products, again, remember this 38 ATP. And these carbon dioxides that were released. Yeah, that was a lot. I'll leave time again for questions if you guys had any you wanted to ask.
Wow.
Yeah, of course.
So are these proteins that are embedded in the membrane, are these the same ones that underwent like prenylation and myelination to bind them to the membrane?
These are in the membrane of mitochondria, not the plasma.
Oh, right, okay, thank you.
All right, so now we just talked about oxidative phosphorylation and the completion of that. What's also important to add is that, again, remember the mitochondria has several membranes or layers, and so also not all of these are permeable. Some of them are large or polar, some of them might be charged. And so you can imagine it might be difficult to transfer them, right? And so what we've adapted to doing is providing these different carriers. And so you can see a lot of these intermediates, you see pyruvate, malate, fumarate, succinate. There's essentially a carrier for a lot of these intermediates that allow them to move compartments of mitochondria as needed, as we move through the TCA cycle and oxidative phosphorylation. Also, just for completeness, you also see ATP, ATP carriers, you see phosphate carriers. This allows again for the movement of these different intermediates and substrates as needed for these different metabolic pathways. So if you remember, I talked about earlier how there's also these amino acids and fats that are playing a role in these metabolic pathways. Here's a perfect example with amino acids. So you can see this is on the right. You can see a lot of these intermediates of the TCA cycle playing out, right? But what's happening is that as we move through these pathways, you also have these amino acids, so like alanine, aspartate, glutamate, so these acidic acids, right? Last two. What's happening is that through these transamination reactions, so the transfer of an amine in place of a carbonyl or vice versa, can sort of interchange. So if we had a pyruvate, for example, we could, through a transamination reaction, give us an alanine or vice versa. And you can imagine this might be especially useful in the case that maybe we see a depletion of one of these intermediates. So for example, if we see a depletion of acyl acetate, one way to remedy this and continue the TCA cycle will be through a transamination reaction. So sort of eliminating this amine group on aspartate and giving us an acyl acetate. And so this second point is kind of just reminding you that as we're moving from aspartate to an intermediate like acyl acetate, what's happening is that we see this catabolic breakdown of the amino acid. However, if you go in the reverse and you have one of these intermediates giving us an aspartate, this again is an anabolic, so sort of a buildup or synthesis of the amino acid. What's also interesting is that with these processes, these are often the alanine, aspartate, and glutamate. These are non-essential amino acids, so they can be produced de novo, so from scratch. But we also have non-essential amino acids, so like phenylalanine, histidine, like these that we need to acquire from our diet, so we can't do reactions like this. So this next slide is really dedicated to now talking about the breakdown of fats. So to kind of summarize, the way that they do this is through beta oxidation. So beta oxidation, again, is breaking down fats, and what it's doing is taking these long chain fatty acids, and through a sequential step process, it's taking away 2 carbons in each reaction, breaking down these fatty acids step by step. But what's sort of important for us is that one of the products of beta oxidation is an acetyl coenzyme A. And if you remember, acetyl coenzyme A is one of the molecules that feed into the TCA cycle. So you can kind of see how beta oxidation of fats can feed into the TCA cycle. And I will go over now kind of like these steps right here. So again, we kind of start with this long chain fatty acid. Here you can see, so the long chain fatty acid here is sort of represented. Here, this coenzyme is just linked to the long chain fatty acid. As we see, we have this dehydrogenase reaction, so we're familiar with that, right? It's forming an FADH2 leucine equivalent. And then in this next step, we see an enol coenzyme A hydratase. So this is a water-dependent process. It's sort of eliminating this double bond, giving us this alcohol right here. And then in the next step, we see another dehydrogenase reaction. So we see the formation of NADH. and the oxidation of this beta carbon. So this alcohol has become a carbonate. So this is kind of where we get the term beta oxidation. And so now the last step here, you see this thiolase enzyme catalyzing this reaction, so this oxidation of this 3-keto-acyl coenzyme A. And what it's doing is it's facilitating the, I'll also add that after we have the oxidation of this alcohol to this carbonyl, I don't know if you guys are taking like organic chemistry, but what this does is make this carbon especially electrophilic. And so what's happening is that this oxygen is especially electronegative. And so it's drawing these electrons away from the carbon, making it more prone to a nucleophilic attack. So what's happening is this coenzyme A with this thiol group, this is acting as the nucleophile here. And then what it's doing is it's facilitating this attack, which breaks this molecule into not only an acyl coenzyme A, but that acetyl coenzyme A as well. And this is how we get acetyl coenzyme A. Does anybody have any questions on that process? So now just kind of talking more about the breakdown of fats, right? So if you're familiar with this triglyceride, this is one of our fat storage molecules. It's made up of this glycerol backbone right here. And these three compartments right here are all these fatty acid chains. So this is similar actually to a lot of our phospholipids. The only difference is that we don't have this polar head group. And so as we break down these triglycerides through like the glycolysis reaction, what happens is that we have this glycerol, right? It's coming off this triglyceride. We also have these fatty acids that are breaking off. And so now we'll talk about how these are like sort of playing a role in a lot of other metabolic pathways. So first with glycerol, if you remember, what can happen is with these glycerol molecules, they can become phosphorylated and form glycerol aldehyde 3-phosphate. So glycerol aldehyde 3-phosphate, if you guys remember, actually I'll post this to you guys. You remember what, this is an intermediate of one of the metabolic pathways we talked about already. You guys remember? Yeah, exactly. Yeah, great. Yeah, so glycerol aldehyde 3-phosphate is one of the intermediates of glycolysis. And so as you can see, the arrow is pointing, essentially saying that we can take this intermediate, again, from the breakdown of these triglycerides and use it for glycolysis. We can also use it for gluconeogenesis as well. So the other thing I mentioned was from these triglycerides, we can take fatty acids. We can generate these into acetyl coenzyme A, remember through beta oxidation. Now from acetyl coenzyme A, if you remember, this is one of the starting molecules for citric acid cycle. And so this is one way that we can feed into another metabolic pathway. But also we can feed into what we call the formation of ketone bodies. So this is another thing we'll talk about in the upcoming slides. It has a lot to do with dieting, and we'll talk about those very soon. Yeah, so that was all I had. Just wanted to thank you guys a ton. This was awesome. Thank you guys for figuring it out. If you guys have any questions, feel free to email me or just come to my office hours. Thank you guys so much. Appreciate it.
No, I don't know. I can come back.